Hi, everyone. Welcome to Foresight Biotech and Health Extension Group, sponsored by 100 Plus Capital. Really excited that you're here. You have been to many of our in-person events, including technical workshops in longevity and our annual vision weekends. So it's really fun to have you here as well to talk a little bit more about your current project, which is activating mitochondrial heat production to reverse obesity. And I've seen some of the slides before. Yeah, I'm really excited for what you'll be discussing today. You're with Equator Therapeutics, and I'll also share a little bit more info on you and on the company in the chat. But without further ado, the stage is yours, and I'll be in the chat monitoring questions.
Yeah, thanks to the Foresight Institute for hosting today. I'm going to tell you about our work at Equator Therapeutics, developing a drug to burn fat through natural heat production. First, I'll give you a little bit of an overview of the obesity field and where it is today to contextualize our work, and then I'll get a bit into our mechanism and supporting data. Yeah, when it comes to health problems, of course, aging is the main cause of morbidity ultimately. But if you look at kind of what drives the most health problems aside from aging, obesity is obesity is the main factor upstream of health conditions. So the costs, both healthcare and economic, of obesity exceed $2 trillion a year. There are a very broad range of phenotypes that are affected. Most of the negative health impact is mediated through the increase in metabolic disorders such as type 2 diabetes, but you also have many other effects. For example, sleep apnea, gastrointestinal reflux. And so reversing obesity has the potential to halt or reverse the progression of these associated health conditions. Now, if you look at the prevalence of obesity, so just focusing in on the United States, the prevalence of obesity has been increasing continually over time. And this paper from 2019 projected obesity rates in 2030, projecting that by 2030, here's what the projected map of obesity across the United States looks like. You see kind of a variation across states ranging from 40% to 60% of the adult population being above the obesity threshold. If you restrict to patients who are of heavier body weight, severe obesity, body mass index 35 and higher, then looking across the states, at the low end is California, where many of us are based. There you're talking about a little more than 15%, whereas if you go to the deep south, you get up to over 30%. And nationwide, just focusing in on this bottom panel here, maybe I'll just grab a laser pointer. So you have this is nationwide, about 50% projected to be above the obesity threshold, about 25% above the severe obesity threshold. When we think about kind of health, a lot of what's most visible to us is what's geographically proximate, and in the Bay Area, we're way below national baseline for obesity prevalence. So stepping outside of our context, you see this showing up as a really major health concern. And if you look at years of life lost due to obesity, so here I'm showing representative data on Caucasian women. The graphs look similar for other demographic groups, a little bit higher for other demographic groups. So what's being shown on the horizontal axis here is body mass index. And on the y-axis is the number of years of life lost associated with being of that body mass index as opposed to healthy body weight. So what is that in the overweight range? You're talking about between 0 and 1 years of life lost, whereas in the kind of what's called class 1 obesity, the first kind of tier of obesity, 1 to 2 years of life lost. And as you pass into severe obesity, you get up to 2 or more years of life lost. With COVID, the highest risk factor for death, aside from age, was metabolic disorders associated with obesity. And if you're able to create a drug to reverse obesity, there are prospects for increasing life expectancy by a couple of years for people in the severe obesity bracket. And it's quite understandable to want more than that, of course. I became interested in this project because it seemed unusually likely to be tractable. When it comes to efforts to increase life expectancy by 15 years, I would be thrilled to do that if I knew how. But yeah, when I was in math grad school, I remember one professor saying, my favorite problem is a problem that I can solve. Here I had some sense for what might be done, which led me to start Equator Therapeutics with my co-founder. I'll get into that a bit later. Yeah, the projections that I gave above were in 2019. Since then, there have been crucial developments that maybe will mitigate the increase that I was gesturing at. So here I'm going to show you the history of FDA-approved anti-obesity drugs. These are drugs that were approved for long-term management of obesity. So yeah, the first goes back to 1999, orlistat. So there you see a pretty small effect size, about 5%. The way that it works is it blocks absorption of fat. One could also put metformin here, perhaps, or acarbose rather. With acarbose, you block it's not officially approved for obesity, but you block intake of starch to some extent, and you can see a modest weight loss with that as well. So yeah, orlistat was the first FDA-approved drug. Then in 2012, 2014, you had a few more come along, glycemia, which can result in about 10% weight loss. You have contrive, which results in about 5%. And then in 2014, you also have the approval of the first GLP-1 receptor agonist. So this is Saxenda. It was developed by Novo Nordisk, and it was initially a type 2 diabetes drug, but then it was found that it causes weight loss as well through appetite suppression. And there you can get up to 10% weight loss. But it wasn't until the most recent generation that kind of people started to become really excited because with the previous ones, the kind of efficacy to tolerability ratio was good enough for people to be taking them sometimes, but it wasn't really striking. And with most recently semaglutide, which is branded as Ozempic for type 2 diabetes, it's branded as Wegovy for obesity. Wegovy was approved for obesity in 2021, and it was the first anti-obesity drug to be reported on to cause 15% weight loss. You've probably seen a lot of news about this drug. Elon Musk endorsed it. There have been concerns about whether it depletes lean mass, muscle mass, which you might also have heard about in the media. But yeah, basically people have been able to lose weight successfully on this drug. And there's been a kind of tide of change on the, payer side, where healthcare systems are willing to cover glutide in large measure. This is the first anti-obesity drug that is reaching a large population, basically. And Eli Lilly has a competitor, tirzepatide, which shows even greater weight loss. It's expected to be approved in 2023, 2024. And basically all of these medications work by reducing energy intake. So it's basically reducing the number of calories that are consumed or reducing the number of calories absorbed. And obesity has been, from a commercial perspective, maybe the largest story in biotech after COVID vaccine, after the focus on COVID vaccines diminished. So if you look just over the past 2.5 years or so, and you look at Novo Nordisk, the company that developed semaglutide, Wegovy, what is that the stock actually increased by over $100 billion just over the past 2.5 years. Novo had not been amongst the largest pharma companies. Now it is. It's in the top 20 stocks by valuation. Eli Lilly, with their competitor, which is looking to be more effective, its stock has gone up by a similar amount as well. So yeah, anti-obesity drugs have generated over $200 billion in shareholder value just over the last 2.5 years, which is pretty striking. So if you look at Wegovy though, Novo's drug, for example, what you find is that the effect size is large in the patient populations that they reported on in their kind of keystone study, but still falls short of reversing obesity for most patients. So the effect is larger in people with type 2 diabetes than without type 2 diabetes than it is in patients with type 2 diabetes. It's also larger in women than it is in men. And but basically for women without type 2 diabetes, on average, you can reverse the first tier of obesity. So like body mass index 30, you can return people to healthy body weight. But as I was showing you at the beginning, a pretty large portion of patients have a need to lose substantially more than that in order to get into the range where less than a year of life is lost. So basically, this is a major advance, but it falls far short of actually fully addressing the problem. Lilly's drug tirzepatide has somewhat larger effect size, but it's still the case that there's a lot of work to be done. And basically, to date, anti-obesity drug development, as I was saying, has mostly focused on reducing caloric intake. So that's how this newest generation of anti-obesity drugs works, by reducing energy intake. And yeah, there's still no FDA-approved therapy for addressing the other side of the equation, for increasing energy expenditure. So that's what we're trying to develop at Equator. And yeah.
About amphetamines.
Brian, was that you?
Yes, it was.
Okay, yeah. How about amphetamines? So amphetamines can increase energy expenditure. They result in people moving around more. When people are stimulated, they move around more. But yeah, if one looks at the 24-hour picture, there tends to be a compensatory crash. Or you can just take them all the time and not sleep at all. But empirically, they're for long, when it comes tolerance, them develops, and empirically, you have not been able to see enough weight loss on them to get FDA approval. Yeah, and where are we coming from? Basically, if you look at the energy production and expenditure of the body, it's mediated through mitochondria in considerable measure. Mitochondria convert food calories into voltage, and then that voltage is used either to power ATP production, which powers physical activity, or it's used to generate heat in order to keep people warm. Now, in modern living conditions, in contrast with ancestral conditions, the fat-burning capacity of natural heat production is inhibited. So our ancestors were exposed to the cold with reasonable high frequency, not all the time, but at night during the winter, if you were some distance from Equator. And so if you think in terms of obesity arising as departure from our ancestral conditions, the things that are most salient are people are eating more and they're exercising less. But there's actually this other thing, which is that people are producing less heat and so have a lower resting metabolic rate on average. And basically, there's this pathway that's not being utilized very much that has effects similar to exercise. And basically, if you can stimulate this mechanism, you stimulate the same metabolic pathways as exercise, and you could have similar health benefits to aerobic exercise. So like exercise, you activate what's called the AMP kinase, which activates these anti-aging pathways and inhibits these pathways that are responsible for aging processes. So activating the AMP kinase, it inhibits mTOR, it inhibits NF-kappa B, it activates the FOXO genes, and so forth. Yeah, you can have a quite beneficial effect similar to the effect of aerobic exercise by activating mitochondrial heat production. That's what we're trying to do at Equator, develop the drug to increase natural heat production. quite slightly, not very much, not enough to make people hot, but enough to slowly and steadily burn calories throughout the day to affect weight loss. Yeah, historically, so why hasn't there been more work, weight loss drugs affecting weight loss via increase in energy expenditure? So there has been work, but what people found is compounds that are highly non-selective. So yeah, people looked at compounds called protanophores, DNP, which I'll be talking about a bit more, highly non-selective, and some other classes of things as well, beta-3 adrenergic receptor agonists, growth hormone, thyroid hormone. And these things will increase resting metabolic rate and burn calories in that way. But you have a side effect profile that basically makes these things not worth pursuing. Yeah, and what this comes down to is that if you look at reactions responsible for heat production in the body, you have some where heat is being produced as a byproduct of an essential physiological process. And you don't want to mess with essential physiological processes. So you can produce heat in this way, but that's not the safe way to produce heat. The safe way to produce heat is by mitochondrial heat production, which is this mechanism that evolved solely for heat production and where activating it does not disrupt essential physiological processes. So yeah, that's what we're up to. Compounds that burn calories through heat production have been known for 100 years. The most famous exemplar is this compound DNP that was discovered in French munitions factories during World War I. Factory workers breathed in the dust, lost weight. It was repurposed as a weight loss drug in the 1930s. It was found to be highly effective. It was used by 100,000 people. Weight loss of 3 pounds a week was standard, but the drug turned out to have prohibitive side effects, and so it was banned by the FDA. But since then, 100 years ago, there's been this dream of, is there some way to do it safely? So yeah, just to dive in a little bit more on DNP, there's a natural mitochondrial heat production mechanism that I was discussing. Food and oxygen are combined to generate voltage across the inner mitochondrial membrane. That voltage can be used for heat production, or it can drive electrical current through a thermogenic protein generating heat. That's what happens physiologically. This compound DNP that was highly effective in the 1930s actually does do that, but it doesn't just do that. So it activates proton currents not only in the inner mitochondrial membrane, but also in other membranes like the plasma membrane and like lysosomes. When it activates proton currents in lysosomes, it interferes with the function of lysosomes, which is autophagy. Autophagy is an anti-aging process. You don't want to be interfering with essential physiological processes in this way. Yeah, and again, this contrasts with natural mechanism where you have long-chain fatty acids activating this thermogenic protein in the inner mitochondrial membrane. So why was more work not done on this before? So yeah, the work identifying and characterizing the protein and the mechanism of action was done for the first time over the past 10 years. So yeah, my co-founder, Yuri Karichak at UCSF, characterized this mechanism. This is a paper from Nature published in 2019. There he characterized the mechanism in most tissues. He had previously given a detailed characterization of the mechanism in brown fat, which is the tissue that people have studied thermogenesis in the most. Adult humans don't have very much brown fat, so you need to activate the mechanism in tissues other than brown fat, like skeletal muscle. Yeah, first there was brown fat, then there was the results in skeletal muscle. Then there was an in-depth study of compounds such as DNP and how they relate to the picture. So that this is the foundational research on which Equator is based. And yeah, my co-founder Yuri's major innovation on the technical side was this one of the number of innovations on the technical side, but the one that I'm highlighting here is this method for directly recording electrical currents through the inner mitochondrial membrane, which had not been possible before Yuri's work, and then he used it for target identification and measurement of target engagement. So this is a physical measurement that's a lot more precise than what you get in the biological sciences for the most part. So it allows for very precise interrogation of the action of compounds. So at Equator, we developed a screening funnel. We would start with some high throughput screens, which I haven't described here, but then compounds that are identified as hits. We test them for whether they induce the thermogenic currents in mitochondria. We test them in the plasma membrane for whether they induce currents there. We don't want currents in other membranes. Then we move them forward to measure thermogenic respiration in cells, and then we move those compounds into animals. So we've done medicinal chemistry optimization on 4 series for developing molecules that more selectively engage the target. First looking for selective target engagement, then improving them for animal use basically, and then moving into animal efficacy. I'll focus in on the first of these. So the first of these is EQ1. What I'm showing you here is this crucial differentiation from DNP. The first 3 bars correspond to currents induced in the inner mitochondrial membrane. So here's DNP as a reference point. You have currents increasing in a dose-dependent fashion. For our compound, you do as well. The next 4 bars are currents measured in the plasma membrane. Now, we measure currents in the plasma membrane at pH 6.5, which is the standard condition. We also measure them at pH 5 in order to model currents in lysosomes where the pH is 5. You don't want any of these currents. So with DNP, you do get these electrical currents in other membranes, whereas for our compound, you're clean in them. Here is a depiction of the increase in oxygen rate consumption in cells. Oxygen rate is proportional to caloric burn rate. So what is that for DNP, you have this increase in a dose-dependent fashion in wild-type cells that have our target protein, and also cells that don't have our target protein. And that in both cases go up. So what we have is a molecule that like DNP increases metabolic rate in cells in a dose-dependent fashion, but we have higher selectivity for our target. So if you look at those cells that are missing our target protein, you have a smaller effect relative to the cells that have the target protein compared with DNP. So if you measure percent dependence on our target, compared with DNP is at about 60%, we are at about 80%. So we've been able to achieve greater target selectivity than DNP. And the most recent data, this came in just earlier this week, so I was scrambling to put together slides. I managed to put together one. So this is in diet-induced obesity mice. And what's being shown here is weight loss in grams. And at 3 ascending doses of DNP, at 3 ascending doses of our compound, the study so far has been 14 days long. This is a 2-week interim report. So we're looking at a decrease in grams as a function of time for 3 doses. And what is that our compound causes about as much weight loss as DNP, perhaps somewhat more at the top 2 doses within error bars, but kind of trending in the direction of greater weight loss than DNP. For DNP, the highest dose tested here shows toxicity. For our compound, there was no toxicity at the highest dose. And these animals are about 44 grams. So we're talking about like 10% weight loss at the highest doses over the course of 2 weeks, which is enough we'll see what continues to happen, but it's enough. It looks like we're trending in the direction of a meaningful clinical effect. And that's where we are as far as this compound goes. But what the safest drug of this type would be a drug that activates the mechanism, but only partially. A drug where no matter how much of the drug you add, you will only increase metabolic rate by a fixed amount. What I was just showing you was a compound that looks to be somewhat safer than DNP, but which still, like DNP, can overstimulate the mechanism. So we want a compound that will not overstimulate the mechanism at any dose in order to achieve a very high ratio of toxic dose to therapeutic dose in order to be able to reach a large patient population. And in a recent breakthrough that I still have to produce slides for, we've identified the first compounds known that stimulate the mechanism only to a mild degree, no matter what the dose. Yeah, but that's where we are. Yeah, naturally, we have funding needs to move this work forward. We've achieved animal efficacy here. We are looking to raise $15 million to develop a candidate, followed by $30 million to bring the candidate to clinical trials. And yeah, right now we're raising a bridge round. So this is on a safe round size of $2 million, a discount of 30% from the Series A valuation, and evaluation cap of $30 million post-money. Yeah, and yeah, just to acknowledge, of course, I'm only one of a number of team members who have made very valuable contributions here. Yeah, my co-founder, Yuri, I mentioned, laid the foundational research and has served as our chief scientific officer. Catherine Widdowson, medicinal chemist with 30 years of experience at GSK. Yeah, and then we have a scientific team, including Sasha Yukari, who I worked with previously at BioAge, who led the preclinical development on BioAge's asset that recently showed positive results in a phase 1b clinical trial preventing muscle atrophy. Yeah, so I'm very fortunate to have collaborators on this who have moved the science forward greatly. And yeah, as far as our investors to date go, one of our first investors, Harrison, at 100 plus capital. Alongside that, we went through Y Combinator. We were awarded a phase 1 SBIR grant to progress our work. SB Lawton Capital, HealthSpan Capital, Samtel Angels, and then, a number of angel investors who have contributed alongside these actors. Thanks very much for your attention. Happy to take questions.
Wonderful. Thank you so much. We already have a lot of different questions.
Okay.
I will be starting with a question from Micah in the chat because he has no mic, and then I'll hand it over to Cosmo and Larry. First one, is the caloric intake of the mice measured or controlled in some way? Were the mice overweight to start or healthy weight?
Oh yeah. These are diet-induced obesity mice. So these are mice that are close to double healthy body weight. And what was the other part of the question?
If the caloric intake of the mice was measured or controlled.
Yes, we did measure caloric intake and saw no increase in caloric intake. So the weight loss is not due to reduced caloric intake, but rather due to increased metabolic rate.
Wonderful. Cosmo, we can't hear you. I'm going to mute you. It's very static. So either you can rejoin or put your question in the chat and I'll ask it for you. Does that sound good? Okay, next one up we have...
Interesting science behind it. The one concern, of course, with these things that induce a little bit of mitochondrial dysfunction is heart failure. And obesity and heart failure sometimes go together. I think there might be a window for therapeutic intervention for these type of compounds, but this is going to cause a problem to induce heart failure because ATP and phosphocreatine are so essential to the functioning of the heart, and not creating enough of that could cause some problems.
Yes. Yeah. So ultimately, basically, if you overactivate our mechanism, you end up having a reduction in ATP production because the voltage is being used for heat production rather than ATP production. And in the heart, there's strong need for ATP. So there's certainly a conceptual issue there. So there are several things to say about this. So one is that our target protein is the same as the protein through which ATP and ADP pass. So when needs for ATP are high, that channel is being used for its primary purpose of transport of ATP and ADP. And so the thermogenic current is blocked. to some measure, to some extent. Oh, now, of course, that's a theoretical argument. You ultimately want to know what happens empirically. So historically for DNP, the toxicity of DNP was mostly not in the heart. So it seems like heart is a tissue where you are actually relatively okay. But yeah, there's a lot more work that's needed here for clarifying the safety profile.
Okay, I'm gonna try to ask Cosmos questions. Are these effects rescued with either carboxychloroquine or bronchinic acid? Wow, that's quite a mouthful of a word. It's also here in the chat.
Yeah, what are those noises? Anyway, yeah, are these effects? I'm not sure which effects you have in mind. We have looked at those compounds in relation to our compounds, but yeah, it might be most efficient to talk offline just to enter again into the details. Yeah.
Okay, wonderful. I'll encourage everyone else to also feel free to continue dropping questions in the chat or raise their hand. Otherwise, I'm going to go ahead with a few of the questions that usually folks are rather interested in here. One is finding out a little bit more about what's immediately ahead for you. So what are like some of the next kind of challenges that you're tackling? Are there any kind of like thoughts for collaboration? Obviously, your fundraising, and I already saw lots of aligned and dear partners in the existing investment team. But yeah, just getting a little bit more of a better idea of what's immediately ahead for you, like in the next year or so, would be really useful.
Yeah. Like I said, this discovery of, so yeah, at the highest level, like when it comes to resources needed, funding is the kind of main limiting factor. But when it comes to activities, this discovery that I alluded to at the end of this is quite recent, of compounds of bounded maximal effect size. When I started the company, I thought, well, first develop a compound that has a worse safety profile and then raise funding and then maybe progress that to clinical trial for some patients and then work on a safer compound after that. But basically with the most recent discoveries, we will be immediately trying to develop these compounds that show only partial activity. As far as those compounds go, we're at the beginning of the process. So there's a lot of kind of medicinal chemistry to be done here.
Okay, awesome. And if someone new was entering the field, like we often have new members joining that are more in the early career stages, and they often just wonder, like, how could they make progress on something like this in the future? Do you have any kind of like outstanding requests that people should be working on, whether or not it's an Equator or perhaps individually?
Yeah, let's see. Do you mean in relation to Equator or in relation to the field as a whole?
Either. Just what are a few kind of headaches that is holding the ship back that you really want solved, either at Equator or by someone else?
Yeah, certainly. If anybody, we do have substantial staffing needs. We also have substantial funding needs, and we need funding to hire people at more or less. But certainly, if anybody who's hearing this is interested in the possibility of working with us, please do reach out to me. We have needs. We have scientific needs. We also have non-scientific needs. We have like in particular, we have operational needs. There's a lot of value to contribute on that front. Yeah, in terms of the field as a whole, one thing that's needed is interpersonal communication skills, because the drug development, it's the most heterogeneous context that I've been in, where you have 5 or 6 disciplines that need to be drawn on in order to successfully get a drug, which means that you're communicating with people who are coming from a variety of cultural backgrounds. There are different implicit assumptions, different language used. And so speaking for myself, the main thing that I see as needed is like ability to engender communication between all of the people involved.
Cool. I have a brief intervention here from Micah. Do you think weight gain associated with aging is caused by reduction in heat production or for other reasons?
Sorry, can you repeat the question?
Yeah, of course. It's also in the chat. Do you think weight gain associated with aging is?
Yeah, it's a multifactorial thing. I wish I could give a comprehensive answer to that. There is a decrease in heat production with age. There are multiple reasons for that. One is that the amount of brown fat that you have, adult humans don't have very much, but it goes down with age above and beyond that. Another thing is that you begin to lose muscle, and part of what drives resting metabolic rate is muscle mass. So with loss of muscle, you have loss of metabolic rate. I think Cosmo probably knows more about this than I do holistically, but yeah, it's a multifactorial thing.
Cool. Cosmo, feel free to drop your comments here in the chat in the absence of voice working well. But yeah, I think in general, could you lay out a projection of if everything went really well, where could the field, like which is rather emerging still, I assume, be in about 5 to maybe even 10 years? What would be like an amazing outcome that would come out of this work? What could people get really excited about if this actually works?
Yeah. So basically, I mentioned the stimulation of the same metabolic pathways as exercise. Even for people for whom obesity is not a problem, stimulating the same metabolic pathways as exercise is still valuable. If you're talking about quite elderly people who have difficulty doing exercise owing to being frail and risk of injury going up, this is something that could give the metabolic benefits of exercise, while being safer in that respect. There's certainly potential for pretty broad spectrum anti-aging benefits of this. And if the better the safety profile we're able to achieve, the more patients could benefit from this. And this could end up being like a staple part of a kind of anti-aging treatment regimen.
Cool. And in terms of safety profile, what ought one worry about there? Is there anything where you're like, ooh, this could potentially be like a challenge that you're facing when it comes to just safety?
Yeah, sure. So the fundamental issue here is that overstimulating, if you produce too much heat, if you use too much of the voltage for heat production, you don't have enough voltage for ATP production. So ATP starts to be produced by glycolysis, producing lactic acid. This is the same as what happens when you do heavy exercise. You produce lactic acid as a byproduct of producing ATP through glycolysis rather than via mitochondria. And then the nature of toxicity is that if you overstimulate the mechanism, then you produce too much lactic acid, which can lead to death. There's a definite safety issue here. The key question is how large a therapeutic index can you achieve? How big a separation can you achieve between the minimal efficacious dose and the toxic dose? And that's what we're working on.
Awesome. Perhaps we have one more from Micah. Can we combine your solution with appetite suppressants?
Yeah, so yes, and indeed the two things are complementary in relation to one another. Because if you suppress appetite, then there will tend to be some downregulation of metabolic rate in order to, like, your body thinks that it's not getting enough energy, and so it tries not to use as much energy. You end up having a kind of reduction in rate of caloric consumption in that way. And so by applying ours in combination with that, you can increase energy intake back to where it was or higher in order to achieve greater combined weight loss.
Awesome. Great. Then my final question would be, if people here are interested in really helping your work along, how can they best contact you?
You can reach me by email. It's my first initial last name, or just my first name, Jonah at equatortherapeutics.com. I dropped it into the chat.
Okay, wonderful. Thank you so much for joining us. And I think, I can just echo Cosmo in the chat, an amazingly awesome talk. So thanks a lot for discussing this. And I hope to see you in person, our next technical workshop, or very, very soon afterwards in the virtual meetings. Thank you, everyone.
Thanks, Allison. Have a good day.